Hello and welcome to our PhysioEx experiment, and that will be exercise 8, the chemical and physical processes of digestion. And we're going to be doing activity 1 of exercise 8, which is assessing starch digestion by salivary amylase. So the objectives are to explain how the enzyme activity can be assessed with enzyme assays, mainly the IKI assay and the Benedict assay. So what's an assay? An assay, we have two sides of a reaction. We have the reactant side and the product side. And between them is the arrow. On top of the arrow is our enzyme. The enzyme is the catalyst that is speeding up the reaction that would otherwise take millions of years to take place. And the enzyme speeds up the reaction by decreasing the activation energy barrier that must be surmounted in order to convert the reactants into products. But how do the assays work? The assays, I must have an assay for each side of my reaction. The IKI assay will target my reactant side, and the Benedict assay will target my product side. So if my IKI reactant converts color from yellow to black, that means there is a lot of reactants. And that means my reaction is not ongoing and I'm unable to convert my reactants into products. But if my Benedict reactant, which is my Benedict assay, my Benedict marker, that is usually blue color, unlike the IKI, which is usually yellowish, the Benedict marker is blue. It converts upon interacting with sugar levels in 3 degrees. If there is a concentration that's high, it will convert color from the blue original color into green. If there is even more sugar, it will convert from blue, but not into green this time. It will convert into orange. So orange means there is even more sugar than green. And if the Benedict assay converts from blue into red, then that's the maximal highest amount of sugar. So if I have 3 test tubes, one is 70 milligrams per deciliter, the other one was 90 milligrams per deciliter, and the third one was 100 milligrams per deciliter, and I apply the Benedict assay, on the one that was 70 milligrams per deciliter, it will turn color green. And on the one that's second degree, that was about, let's say, was 80 milligrams per deciliter, more than the 70, it'll turn, for example, into orange. And then the last one, if it's 100 and above, now it's going to turn into red. Then we're going to define an enzyme. And we know what an enzyme is. It's a catalyst that is usually not, that is always not consumed in the reaction. It only creates modification. It takes into its active site the reactants and causes chemical conversions and breaks the bonds and rearranges all reactants into products. And it's a catalyst that means it will speed up the reaction by decreasing the activation energy barrier. And hydrolase, that means it will hydrolyze. It will utilize water to cleave the bonds. And a substrate is a reactant. And a product a substrate is converted into a product. And a control, we can have a positive control that once I add the positive control into my reactant, I will have a known outcome, a known positive outcome. And if I add a negative control, I will have a negative response, meaning for example, if I have no sugar in my test tubes, that's a negative control because once I apply the marker, it will not change color. It will remain that same color. But if I have a known product, and that would be an example of a positive control. To understand the specificity of the amylase action. So we know that amylase will target carbohydrates and starch. So starch is a maximal storage form of carbohydrates. So we have a lot of carbohydrates that are linked by a alpha 1,6 linkage, and that's amylase, while cellulose is beta 1,6 linkage. So amylase is going to cleave the bonds that connect for me amylase, that connect for me starch, and convert it into these disaccharides, known as the maltose. And then from maltose, maltose is made up of 2 glucoses, and that will give me glucose. And then we're going to perform the appropriate chemical test to determine whether digestion of a particular food has occurred. Then we're going to discuss the possible effect of temperature and pH on amylase. And we know obviously that enzymes, being proteins, they are very sensitive and susceptible to temperature. Very high temperatures will cause the proteins, these amino acids, to denature and break down. While freezing, we utilize freezing to store these enzymes in the fridge. So usually their activity will not be affected. In fact, we keep them stored frozen. But if you froze them for a very long time with a very severe freezing temperature, that will also cause them to disintegrate. So moderate freezing is favorable. And the pH, there's an optimal pH for the amylase to work, and that is usually around the pH of 7, which is found in the mouth. While pH of 2 in the stomach, that's too acidic. That is not optimal and will inhibit the will deteriorate and causes a very low activity of the enzyme that could be proven by a drop in the products formed. And the reactants will accumulate, but the products will go down because the pH is not optimal, is not supporting my enzyme activity. And the intestines have a pH of 9, and that is also above that optimal physiological pH of 7 that's in the mouth for amylase. That's why amylase is in the mouth. Okay, so that's my reaction, starch plus water. We add to the amylase, and amylase is going to convert for me starch into maltose. Let's do now. We talked about the enzyme assays. So now let's do the pre-lab. The substrate for amylase. So obviously amylase will take starch and other carbohydrates and convert them into maltose and glucose. So the substrate is going to be maltose. No, it's going to be carbohydrates and starch. That sounds like the right answer. Which of the following is true of enzymes? They are used up in their reaction. No, they're not. They become part of the product. No, they're not. They will not. Their activity can be affected by temperature and the pH, of course. Read no further. The reagent IKI tests for the presence of the reactant, which is the starch and carbohydrates. Which of the following is not true of the controls? With a negative control, a negative result is expected. That is true. With a positive control, a positive result is expected. That is true. A positive result with a negative control could mean that a contamination substance is present. That is true. So that means I have a negative control that is not supposed to give me products. I'm supposed to get zero products, and I do have products. That means there is a contamination or a reagent that's contaminated. A negative result with a positive control is required to validate the test. That's not true. It is in fact a positive result with a positive control that is required to validate the test. Which of the following is an end product of starch digestion? The end product of starch digestion is going to be maltose and glucose. Hydrolases are enzymes that break down large molecules into smaller subunits through the addition of water. OH on one side and H on the other side, and that's going to cleave the bond for me. Press submit. Okay, so that's the experiment. So what we're required to do is we're going to drag the test tube to the first holder, and then the others are going to follow through. And once I have 8 test tubes, I have to prepare the solutions. So I have my details. Okay, so pH 7 simulates the optimal pH, homeostatic pH. Actually, homeostatic pH is 7.35 up to 7.45. But pH 7, that's oral pH. pH 7 buffer approximates the pH in the mouth, while pH 2 is in the stomach. The pH 2 buffer approximates the pH in the stomach. And tube 8 amylase starch, pH 9, the buffer, pH 9 buffer approximates the pH in the small intestine. So that resembles the intestine. I have 8 tubes here. I have to make each tube with these content. So tube number 1 is amylase, starch, and pH buffer 7. So take 1 drop of amylase, and then a drop of 3 drops of starch, and then buffer pH 7. So here I only have the enzyme, and I have the reactant. I do not have the product. So now when I remove this assay cabinet, and then now they will interact, because amylase and starch will start interacting, and then products will be formed. So now let's look at two amylase, starch, and pH 7. Amylase, starch, and pH 7. So that is identical to tube 1. So what's the difference? Why is tube 1 amylase, starch, and pH 7 buffer? Amylase, starch, and pH 7 buffer on 2. How about 3 is also the same? It's amylase, starch, amylase, starch, and pH 7. So what's the point of having 3 identical test tubes? And here we're going to be testing the effect of the temperature. So now we have the same pH, we have the same reagent, and we have the same enzyme. Now, one is we're going to keep it at optimal temperature. The other one, we're going to make boiling temperature. And the other one, we're going to make freezing temperature and allow the 3 to run. Then we're going to look at the effects. What happened in the same reaction at physiological temperature, 37 degrees Celsius? What happened to the same reaction, but now no longer at physiological temperature, 37 degrees Celsius, but rather at boiling temperature, 100 degrees Celsius? And then we're going to look at the same reaction, but this time no longer at physiological temperature, 37 degrees Celsius, and no longer at the boiling temperature of 100 degrees Celsius, but now rather at freezing temperature of 0 degrees Celsius. That's for the first 3 tubes. Let's look at tube 4. It says amylase, so drop amylase. Then take deionized water, and that is just to bring up the level of the reagent. And then add pH buffer 7. Well, isn't that interesting? What do we have? We have the enzyme here, but no reactants and no products. So I expect that tube 4 will not change in either color. If I apply IKI, it will not change color because there is no reactant. And if I add the Benedict, it will also not change in color because there is no product. For tube 1, when I'm at physiological temperature, I should have my IKI reagent turn from yellow to black because I do have reactants, but I have no products. So when I have products, I didn't start up with a product because you don't start with a product. You start with a reactant. What's going to happen here is that if, because remember we're testing temperature also, if the temperature is optimal, I will have both colors. The yellow color turning into black for my IKI because I do have a reactant. And once they both interact in the test tube at that optimal temperature, I will have a considerable amount of product being the maltose and glucose. And the color of my Benedict now will increase. So it will change from blue into green. It will change from blue into orange, from blue into red. It will change from blue into red, meaning there is so much sugar, so much product that has been made, reflecting that we are now at optimal temperature. But if we are at a non-favorable temperature, we will have a reaction in the end. We will have reactants turning into products. But when we assess, it will never be from blue to red because that signifies that I do have an enzyme operating at optimal capacity, but here I don't. So it will be from blue to, for example, green. So there's just a little bit of product formed. Let's move on to tube number 5. Here we have deionized water, starch, and pH buffer 7. And tube number 6, I have deionized water, I have maltose, I have pH 7. I have tube 7 being amylase and starch and pH 2. So now I'm playing with the pH factor. One time I am assessing my temperature factor, and the other time I'm assessing my pH factor. Tube 8, amylase, starch, and pH 9. So tube 8, I have amylase, starch, and pH 9 both. Then I'm supposed to incubate them. Descend into the incubation unit. All the others should select the number 1. Should remain in the raised position. That happened. Then select boil. So now test tube 1, we're going to boil it. We have boiled it down, and now the reaction has finished. Then put the number 2. And then for number 2, we want to freeze now. So now we're testing temperature effect. So this one has been boiled, this one has been frozen. Then we're going to incubate. We're going to incubate them, and now they are incubating hypothetically for a whole hour. But this is going to be sped up to like 10 seconds. But we're agitating, everything is being agitated. And so now we are ready to test all of these reactions that are happening here. So what effect do you think boiling and freezing will have on the activity of the amylase? I know that boiling will definitely decrease the amylase activity because it will denature the proteins, protein structure, and denature and destroy the enzymes. But it will have no effect. The freezing will have no effect on the activity of the amylase because remember, we freeze up these enzymes on the fridge to preserve them. But obviously, this is not extreme temperature. This is just freezing. We're not excessively freezing. Now we're going to be using the assays right here. Okay, we're gonna drag the first tube into the incubation unit to the first small assay tube. So we're gonna take a fraction of that, and the rest is gonna go back. And we're gonna test both, one with the IKI and the other one with the Benedict test. The IKI dropper cap. To drag the IKI dropper cap to the first assay to dispense the tip. Okay, so now we're dropping IKI. You see it's yellow. If it turns black, that means there it detected reactants. If it stays yellow, there is no reactant. So right off the bat, you know, tube 1 did have definitely starch. Tube 5 definitely had starch. Tube 7 and 8 definitely had starch. And tubes 2, 3, 4, and 6 had no reagent, no reactant, no starch. Then we're going to do the same thing for the Benedict, but on the other half. So we split our reactant mixture. One half to test for reactants with IKI, and one half to test with products with Benedicts. Okay, inspect the tubes for color change. A blue-black color indicates a positive starch test. Yes, if starch is not present, the mixture will look like diluted IKI. We talked about that. Now let's do record data. So now we have recorded all the data. Okay, stop and think. Tube 2 that had amylase, it has our enzyme, it has our reactant, and that's the optimal pH. Appears to have the same amount of starch digested as tube 3. Amylase, starch, and pH 7. So tube 3, what did tube 3 have? Where's our yeah, so tube 3. Tube 3, the only reagent. So let's compare tubes 2 and 3. So both had amylase, both had starch, both had optimal pH. Only difference was tube 2 was frozen, and the tube 3 was optimal. And they both had the same temperature, and the reaction took place at the same temperature and had the same time, and the products were the same. So that shows you that freezing did not affect the capacity of the function. So freezing had no effect on the enzyme. Perfect. We did drag the Benedict reagent dropper cap. We did that. Select boil. The entire tube rack will descend into the incubation unit and automatically boil. Okay, everything has been boiled. Select the record data. Now we're done. So now let's go and we recorded the data. So now we're done. Let's submit, and now let's compare. Tube 3 had the amylase, the enzyme, starch, the reactant, and the optimal pH 7. Tube 7 had our enzyme. It had our starch reactant, but the pH was 2. It is no longer optimal. So the capacity of the amylase enzyme operating in a reaction under these conditions in tube 7 is obviously going to be lower than tube 3. And the same thing in 9 is also going to be lower. So it says that means that amylase activity was the highest at 7. So let's look at tube 3. Tube 3 had the high 2 pluses compared to tube 7 that had 1 plus and tube 8 that had 1 plus. So that shows you at a buffer pH of 7 compared to 2 and 9, amylase functioned optimally, the best. This activity includes a number of negative control. A negative control is something that when I add it, it will give me negative results. It will deteriorate things. Which tube indicates that the amylase solution was not contaminated with maltose? So in order to be contaminated with maltose, that means my Benedict reagent that detects maltose should never turn from blue into any other color. Okay, and this activity gives a number of negative results, which tube indicates that the amylase solution was not contaminated with maltose. That means obviously I should have a pH buffer 7, and I should have my enzyme in there. Otherwise, I will not be able to have a reaction and deionized water. So that means I must have amylase, and I must not have, if I have amylase, I must not have starch, because amylase with starch will give me maltose, and that will be detected by Benedict. So 4 sounds like the good answer to me, because which indicates that the amylase solution was not contaminated with maltose. That means in this tube here, this will show that there is no contamination with maltose, meaning that if I add amylase with water, there is no starch, so I'm never going to have maltose. So in this reaction, if the Benedict turned from blue to any other color, that means I have a contamination with maltose or glucose, because I had no reactant. Where did the product come from? I had no starch. Where did the maltose come from? Who did amylase interact with? So that means there's a contamination. So that sounds good for me. But if the case was tube 1, that means I have amylase and starch. Then definitely I can never tell if I had a contamination because I have the reactant. So it's definitely not 1. Deionized water and maltose. If I have maltose, the reagent will turn from blue into green. So definitely not 6. And tube 7, amylase and starch, that will give me also the maltose and glucose, and that will cause the Benedict reagent to change color. I don't want all of that. I do not want a change of Benedict. I want to put Benedict as blue and see it as blue. And that will be my control to prove to me that this solution had no maltose. So in order to not have maltose, I must ensure there is no maltose and there is no reagent that amylase, but I must have my enzyme. So I have the enzyme, but nobody to react with it, and ensure that that's how. So it's tube 4. Which tubes indicate that the deionized water did not contain contaminating starch or maltose? So in order to ensure that I had no starch, the first tube must, whenever I add IKI to it, I should only have it yellow. I add it as yellow and it stays yellow. It does not change from yellow to black. And that will only happen if I have amylase, deionized water, and pH 7. That sounds good for me. That sounds good for me because I have no reagent, no reactant. And to ensure that there's no maltose, I should have deionized water, and I should have amylase, deionized water, and pH 7. Like here, yes, so those two. I must have amylase. uh, I must have amylase, like here, amylase, and I must have deionized water, pH 7. That to ensure for me there is no contamination with 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 starch or maltose. 5 will ensure, 4 for me will show that whenever I add IKI as yellow, it will remain yellow because there is no reactant. And whenever I add the Benedict, come in as blue and stays blue, ensures for me there is no maltose. For 5, if I have deionized water and starch and pH 7, and there's no enzyme, that will ensure for me that is a positive control, and that will ensure for me that there is a positive control. That means the IKI will be added as yellow and it will convert into black. But there will be no reaction for maltose because there is no enzyme. And the other one, there is maltose as a product, but there is no enzyme. So this is the positive. Tube 6 is a positive control for me to detect that there is a product or a Benedict turning from blue to whatever. That's a positive control. And number 5 is a positive control for my reactants. And this is the negative control for both. Well, that sounds like a good answer. Explain where and why salivary amylase would be most active. Obviously, salivary amylase would be active in the mouth, because pH 7 is where its peak activity was. Yes, pH 7 was the peak activity for amylase. It gave me plus plus. Amylase never gave me double pluses. Double pluses. Whenever I had amylase, it never gave double pluses except when it was on pH 7. Submit. Now list the substrate and the subunit product of amylase. I know the substrates are starch and the products are the substrates are starch and carbohydrate, and my products are definitely my maltose and maltose and glucose. Okay, what effect did boiling have on the enzyme? It will denature the protein, denature the structure of the protein. And the enzyme has an optimal temperature. Where did the result this compare with your prediction? It will decrease amylase activity, yes, but freezing is actually favorable to store the enzyme. Okay, at what pH was the amylase activity the most active? pH 7, obviously. Briefly describe the need for controls and give an example. So I validate my experiment when my positive control gives me a positive result. And I rule out contaminations when my negative control gives me a negative result. Describe the significance of using a 37 degrees Celsius incubation temperature to test salivary amylase activity, because that's the optimal temperature for the enzyme to operate. That is the physiological bodily temperature, and that is so enzymes operate. Enzymes do not all operate on the physiological temperature, but they have their own temperature and their own pH in order to, so some enzymes work at acidic pH, other enzymes work at basic pH, other enzymes work at physiological pH. This amylase, salivary amylase, so happened to be activated at pH 7, even a little less than physiological pH, which is 7.35. And it also happened coincidentally to work optimally at a pH of 7 and a temperature of 37 degrees Celsius, which is physiological temperature. So that is just to signify for you that enzymes have an optimal pH and optimal temperature to work at. Let's submit and let's, yeah, our answers were correct for the post lab. And I'll see you in our next lab. Thank you very much. Have a nice one.